Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 9 from our Genetics Essentials Concepts and Connections textbook, 4th edition, which deals with DNA replication and recombination. So let's go ahead and get started. Before we get started though, remember this should be a review, a lot of this should be a review of Biology 1406, so I'm going to throw a card up above for my Biology 1406 lecture, which breaks this down in very simple terms. What's happening during DNA replication. But let's touch on it again in this chapter. So remember that eukaryotic cells undergo DNA replication a little bit different than prokaryotic cells. Prokaryotic cells have one circularized chromosome with a single origin of replication, and at that point you have a replication bubble with two replication forks heading in opposite directions during replication of the DNA. In eukaryotic cells, during S phase of interphase, you have replication of the DNA. This is where you copy each and every chromosome and you end up with the sister chromatids. And remember, in eukaryotes, there are thousands of origins of replication, not just one. And each origin of replications, replicon meaning segment of DNA that undergo replication, each replicon is about 200,000 and 300,000 base pairs in length. So you can see here, here's a chromosome, or a segment of a chromosome, I should say. Actually, there would be hundreds or thousands of these origins along the entire chromosome. But you have origin 1, 2, and 3 here represented. Each chromosome contains numerous origins. And then at each origin, the DNA unwinds, producing what's known as the replication bubble. And don't forget, in the replication bubble, you have a replication fork at this end and a replication fork at that end. So replication is occurring in both directions at the same time in each bubble. So number 3 says DNA synthesis takes place on both strands at each end of the bubble as the replication forks proceed outwards. So you notice how the replication bubbles are heading towards each other until they bump into one another. And then eventually the forks do bump into one another. So the forks of adjacent bubbles run into one another and the segments of the DNA fuse. And this is how it becomes complete, producing two identical complete linear DNA molecules. So conclusion here would be the products of eukaryotic DNA replication are two linear DNA molecules. You see that? There's just so much DNA in a eukaryote, you can't have just one origin of replication and finish any day soon. And what do you need for replication? You need a template to copy, a template strand. Remember, out of a double-stranded DNA, one strand serves as the template strand for copying. Then the raw materials you'll need are some dNTPs. Remember, we talked about this before. These are the deoxynucleotide triphosphates, right? These are the building blocks, the C's, G's, A's, and T's for DNA. And then you need enzymes, including the polymerase. This is the enzyme that links those nucleotides together with phosphodiester bonds to make the strands of DNA. And here you can see captured, you have the template strand of DNA, 5' end on this side of the template strand, 3' end on this side of the template strand. Notice how the new strand, called the daughter strand, is being formed. You can see the arrow going down. The daughter strand is always synthesized, you need to know this, in a 5' to 3' direction. So remember, the 5' has the phosphate group overhang. The DNA is always synthesized in a 5' to 3' direction with respect to the daughter strand of DNA. And it's polymerase that does that job. And polymerase, what it does is it takes those dNTPs like ATP, GTP, CTP, TTP. It takes those nucleotide triphosphates, it cleaves off two of the phosphates, and as a result uses that energy to link the nucleotides together with phosphodiester bonds. So a phosphodiester bond forms between the two nucleotides, and that's how DNA synthesis occurs. You need to know that DNA synthesis occurs in a 5' to 3' direction. And again, polymerase adds nucleotides only to the 3' end of a growing strand on the daughter strand. This means that replication can only go 5' to 3'. And remember this from Biology 1406. One strand of DNA is called the leading strand, where DNA replication is continuous because DNA can copy 5' to 3' continuously. But on the other strand of the DNA, because DNA is antiparallel, that strand is called discontinuous because you have to build backwards. And remember, that's where these fragments come into play, called the Okazaki fragments. Hopefully that's a good review for you from Biology 1406. Again, watch that video of my explanation from Biology 1406. If you don't recall how Okazaki fragments work, that video is very good. So here in figure 9.7, you can see DNA synthesis takes place on opposite directions on two DNA template strands. See, in gray, you can see this is the double-strand DNA in gray that you're copying. You have split the two strands apart. The bottom strand is what's known as the leading strand because the daughter strand is being synthesized 5 to 3. This means the more the replication fork unwinds the DNA, the more the daughter strand can copy continuously toward the fork. The leading strand is copying towards the fork continuously, and that's why it's called the leading strand. But on the lagging strand, the DNA is being synthesized 5' to 3' away from the replication fork. This means that it's going to have to occur in segments. You start a segment, you unwind the DNA, and then you'll start another segment, and then you'll have to unwind more DNA to start another segment. So these are what's known as fragments or Okazaki fragments. Again, the leading strand undergoes continuous replication, the lagging strand undergoes discontinuous replication, and those fragments that form due to the discontinuous replication are called the Okazaki fragments. These are discontinuously synthesized short DNA fragments on the lagging strand. And again, please watch my video from Biology 1406 if you need a refresher, a good refresher on how the Okazaki fragments work, because this should be repeat for you. This should be review for you. So discontinuous replication is as a result of which property of DNA? Why do we end up with discontinuous replication? Why do we end up with Okazaki fragments instead of just two leading strands? Why do we have a leading strand and a lagging strand? Well, this is due to the antiparallel nature of DNA strands. Now, let's take a look here more closely to DNA synthesis as it occurs. So again, you've split the DNA. These are the template strands in gray, right? The template is the gray double-strand DNA. The bottom strand serves as the leading strand. The top strand serves as the lagging strand. And here it says on the lower template strand, DNA synthesis proceeds continuously in the 5' to 3' direction, the same as that of unwinding, right? So that's the leading strand on the bottom. The more you unwind the template strands, the more you can copy towards the fork. Okay, but then later on it says on the upper template strand, this upper template strand, DNA synthesis begins at the fork and proceeds in the direction opposite that of unwinding. So it soon runs out of template. So on the top strand, because it's building 5' to 3' away from the fork, DNA synthesis started here at the fork and it built away, and it's going to run out of room. It's going to run out of runway, which means that the DNA needs to unwind more before a new fragment can form on the top strand. That would be the lagging strand, right? So 3, it says DNA synthesis starts again on the upper strand, which is known as the lagging strand at the fork each time proceeding away from the fork. See? So every time you unwind the DNA a little bit, you're going to get another little segment formed on the lagging strand. This is due to the antiparallel nature of DNA. Number 4, DNA synthesis on this strand is discontinuous. Short fragments of DNA produced by discontinuous synthesis are called Okazaki fragments. So you see you've got an Okazaki fragment, but on the leading strand, you don't get these fragments. You copy the DNA, and the more you unwind the DNA, the more you can just keep copying the DNA. Now, in bacteria, replication of DNA occurs a little bit differently than in eukarya. Eukarya, it's the same principles, but there are more players involved. Okay, you don't need to know all the players, but we'll get there. With bacteria, you have initiation of replication at one site. Remember, a single origin of replication called oriC, and then there's a DnaA in E. coli. This is the initiation protein. You need the initiation protein to bind to the origin of replication in order to recruit helicase and in order to start DNA replication. And from there, DNA helicase will bind, and this is part of unwinding. And you'll need to know this for the exam. What are these components of unwinding of the DNA at the replication? DNA helicase unwinds the DNA. The single-strand binding proteins keep the DNA apart, and then DNA gyrase makes sure that supercoiling doesn't occur in front of the replication fork. So you can see here a graphic of this. You've got the oriC, origin of replication. You recruit the DnaA initiation proteins, which then recruits helicase, which unwinds the DNA. And then you have helicase moving in both directions. So this is known as the replication bubble, and you've got helicase moving in both directions away from one another in two directions to copy that replicon. And again, here you have DNA helicase activity, unwinding DNA by binding to the lagging strand template at each replication fork and moving in a 5' to 3' direction. So that's what helicase does. Here's a replication fork. Here's a replication fork. This is the origin of replication here at the center. This is known as a replication bubble. And you have this helicase moving in this direction to the right, and this helicase moving in this direction to the left simultaneously. And it is those helicases that are bound to the lagging strand while they move. Now this would represent gyrase, right? Gyrase, which is a topoisomerase, preventing the overcoiling of DNA. Remember we talked about how DNA has to have 5 base pairs per turn, per 360-degree turn. Well, the activity of helicase introduces more coiling into the DNA, which needs to be relieved by gyrase, the topoisomerase, which relieves the supercoiling. If you don't relieve the supercoiling, you're not going to be allowed, a helicase is not going to be allowed to copy for long. So place these in order. Place the following components in the order in which they first are used in the course of replication. Helicase, single-strand binding protein, DNA gyrase, and initiator proteins. Okay, so remember I said DnaA, the initiator proteins have to bind first, and then the single-strand binding proteins have to keep the two strands apart. Helicase, actually no, the initiator proteins initiate replication, that they recruit helicase. Helicase is what actually unwinds the DNA, and then the single strand binding proteins need to keep the DNA apart, and then gyrase actually keeps the DNA from twisting and coiling too much, supercoiling. So let's see if that's correct. Initiator proteins, yes. Helicase next. Single strand binding proteins next. And then DNA gyrase following that. That's great. Okay, so let's keep going. Now, elongation, this is during, in the process of copying the DNA. What you need are primers. Primers are a group of RNA, a stretch of RNA, a short stretch of RNA. So an existing group of RNA nucleotides with a free 3' hydroxyl group to which new nucleotide can be added. It's usually 10 to 12 nucleotide long, and it's put down by an enzyme called Primase, which is essentially an RNA polymerase. So again, if you don't recall this concept from Biology 1406, please watch that video I posted in the card earlier in this video, because I explained to you that DNA copying cannot occur without a RNA primer laid down first. In order to copy DNA from a template to a daughter, you can't just copy from scratch. You need to first put down an RNA primer and then feed off of the 3' end of that, feed off of the 3' end of that RNA primer in order to copy the DNA. Okay, so RNA polymerase has to first copy a template strand into RNA, a short stretch of RNA, 10 to 12 base pairs long, and then DNA polymerase is able to copy the rest of the DNA into DNA complementary DNA. So let's look at that process here. You have the origin of replication, and then from there you have primase synthesizing that short stretch of RNA I told you about, the primer I told you about, and then the 3' group to which the DNA polymerase can add the DNA nucleotide. So again, when you're copying DNA, the very first thing you need to do is copy the DNA to RNA for 10 to 12 nucleotides. The enzyme Primase takes care of this. It copies the DNA into complementary RNA for only 10 to 12 nucleotides, and that's called your primer. That stretch of RNA is called your primer, and then DNA is allowed to DNA copying is allowed to take place from there on with DNA polymerase. DNA polymerase can then build off of that RNA primer. Again, if all of this is gibberish to you, please watch that video from Biology 1406. You should have learned this in Biology 1406, and that'll make it more clear. But this is taking, we're kind of trying to use that as a launch pad here. So I'm not going to re-explain all that here. So again, you have your primer put down, then DNA polymerase puts down DNA. On the leading strand, where replication is continuous, you only need one primer, and then the rest is synthesized by DNA polymerase. On the lagging strand, however, every time you unwind some more DNA, you need to put down a new primer with new polymerase or primase, and then DNA polymerase will take up from there, or pick up from there. So what you're going to end up in on the lagging strand, you're going to end up with short primers followed by DNA, another short primer followed by DNA. So what you end up with your Okazaki fragments is a bunch of primers followed by DNA. On the leading strand, you only have one primer and then a continuous copying of DNA. On the leading strand, you only have one primer and then lots and lots of DNA. On the lagging strand, you have multiple primers with short fragments of DNA. Those are called the Okazaki fragments. So here, concept check number 4 time. Primers are synthesized where on the lagging strand? Is it only at the 5' end of the newly synthesized strand? The 5' end of the newly synthesized strand. That doesn't make sense. At the 3' end of the newly synthesized strand? At the beginning of every Okazaki fragment? Yeah, that makes the most sense. At multiple places within an Okazaki fragment? No, I think it's C. At the beginning of every Okazaki fragment, you need a primer. Otherwise, you can't copy the DNA. You need to build off of that primer. Okay, so again, elongation, this means copying of the DNA is carried out by DNA polymerase III in bacteria, in E. coli. Removal of the primers, so after you've used the primers to make the Okazaki fragments, at that point you need to remove the primers and replace that RNA with DNA, don't you? Well, that's the job of DNA polymerase I. DNA polymerase I's job is to remove the primers after the Okazaki fragment is formed and replace that DNA, or replace that RNA with DNA. And then ultimately termination occurs when two replication forks are met, when they meet. Again, what does DNA polymerase I do? It builds 5 to 3. What it really does is removes and replaces primers. Okay, removes and replaces primers. And then DNA polymerase III, what it does is it elongates the DNA. It copies the DNA. Okay, so you can see here in this picture, DNA ligase's job. So what happens is when a primer is removed by DNA polymerase I, there is still a nick between this Okazaki fragment on the right and this Okazaki fragment on the left. There's a gap here. There's a nick in the phosphodiester backbone. The two Okazaki fragments need to be linked to one another, and the job of that is ligase, DNA ligase. Ligase does the job of linking this Okazaki fragment to that Okazaki fragment. So again, after Okazaki fragments are formed, DNA polymerase I removes the primers, but it leaves a break in the sugar phosphate backbone between the two Okazaki fragments, and that is repaired by the enzyme ligase. Again, if all of this is gibberish, please watch that video I linked earlier to my Biology 1406 lecture that makes clear of the roles of polymerase I, polymerase III, ligase, Okazaki fragments, leading strand, lagging strand. All of that is clearly explained in Biology 1406 and should be reviewed for you. Again, after DNA polymerase I does its job, the Okazaki fragments are not linked to one another. There's still a link in the backbone of the DNA, which means in the sugar phosphate backbone, there's still a phosphodiester bond that needs to be repaired. And the job of that is ligase. Ligase has that task. DNA ligase fuses. Let's see what it says here. DNA ligase seals the nick with a phosphodiester bond between the 5' phosphate group of the initial nucleotide added by DNA polymerase III and the 3' hydroxyl group of the final nucleotide added by DNA polymerase I. Again, all of the players I just mentioned are listed here on this table 9.3. Please brush up on that for for your understanding and for the exam. And that leads us to concept check number 5, which is which bacterial enzyme removes the primers. Remember that? Which one's job is to remove the primer? Let's go through. Is it Primase? No, Primase's job is to put down the primers. Is it DNA polymerase I? Yeah, actually, DNA polymerase I, its job is to come in after the Okazaki fragment is formed and to remove the primers that are no longer needed and replace those primers with DNA. Is it DNA polymerase III? No, DNA polymerase III's job is to do a majority of the copying of the DNA synthesis. Is it ligase? No, ligase's job is to link Okazaki fragments together by sealing the nick by forming a phosphodiester bond. So it should be B. Yes, B is the correct answer. So in this image, you're going to see how DNA is actually copied when you've got the leading and the lagging strands. So during DNA replication in E. coli, the two units of DNA polymerase III are connected. They're actually kind of connected. And this is a nice video here. If you want to go and see this process, you don't need to know this in great detail for the exam, but you might want to watch this video on YouTube because it's a good video that shows you this process in action. What actually happens during DNA replication is there's a looping on the lagging strand, okay, and the reason for that is because of how the machinery works during replication, which you don't need to know in great detail from my class, but it is really fascinating to watch. So maybe watch that video to understand how the DNA machinery really works and why this looping out happens on the lagging strand during replication. So then let's talk about the fidelity of DNA replication. This means how well the DNA is copied without making mistakes. And the answer is phenomenally well. The DNA machinery, the DNA polymerase, makes very infrequent mistakes. And the reason for that is because it has proofreading ability. DNA polymerase has a 5' to 3' exonuclease activity that removes incorrectly paired nucleotides. If the DNA is being copied and that DNA is making mistakes, if the DNA polymerase makes a mistake, so instead of pairing an A with a T, what if it pairs an A with a G? It has the opportunity to notice that mistake, basically proofread its work. And remember, DNA polymerase III is normally working in a 5' to 3' fashion. It can notice that it made a mistake. It says, oh, I made a mistake. I put an A with a G instead of an A with a T. It can then back up, like throw on the reverse lights and back up, not in a 5' to 3' fashion, but then 3' to 5'. That's why they call it 3' to 5' exonuclease activity. That means it could move backwards and cut out the mistake, cut out that G, and replace it with the correct nucleotide T, and move on, right? So isn't that interesting? Because DNA polymerase has this proofreading ability, this 3' to 5' exonuclease activity, it's able to repair mistakes that it makes, and that is called mismatch repair. Corrects errors after replication is complete, and that is one of the reasons why there's high fidelity when you're copying the DNA. It's very accurate. That doesn't mean that DNA polymerase doesn't make mistakes. It reduces the number to a very, very, very low number. Okay, but it does still make mistakes, don't get me wrong. DNA polymerase III can still make mistakes. It's just really, really infrequent. So then how is this different from eukaryotic DNA replication? Well, in eukaryotes, you have what's known as the origin recognition complex, or ORC, which binds to an origin or replication to initiate DNA replication, or Replicon, I should say. The licensing of DNA replication by the replication licensing factor, these are proteins, when it attaches to the origin. So you have the ORC, you have the licensing factors involved as well. So you've got added components to DNA replication. But keep in mind that you have multiple origins of replication here. And with DNA replication, you have multiple different DNA polymerases, whereas in prokaryotes, you only really have a handful of different DNA polymerases. DNA polymerase I, II, and III. In eukaryotes, you have a host of different polymerases. So basically, you have different players, but the same basic concepts, right? The same basic concepts. So again, concept check. What are some differences from prokaryotes in the genome structure of eukaryotic cells that affect how replication takes place? So what are some differences between prokaryotes and eukaryotes? Obviously, prokaryotes, circularized DNA, not that much DNA to copy, one origin of replication. Eukaryotes, tons of DNA to copy, linearized DNA. You've got the telomeres to deal with, and you've got multiple origins of replication. Let's see what your book says. The size, the size of the eukaryotic genomes is much greater. The linear structure of the eukaryotic chromosomes is a problem. And then you have the association of the DNA with histone proteins. That's right, remember, DNA has histone proteins, whereas bacteria have histone-like proteins, which are less of a problem. Remember, archaea do have histone proteins, though. Archaea do have histone proteins, some of them. So take a look here. These are eukaryotic cells, polymerase, right? Polymerase and eukaryotic cells. You have polymerase alpha, you have delta, epsilon, gamma, zeta. I mean, it just keeps going on and on. So it just confounds the problem. I don't need you to know the difference between all the different types of polymerase in eukaryotic. DNA replication, but just to give you a taste, alpha polymerase is involved in initiation of nuclear DNA synthesis and DNA repair. It also has primase activity. So this is your primase. You have polymerase delta. This one is involved in lagging strand synthesis, whereas epsilon is on leading strand. Epsilon's on the leading strand, delta's on the lagging strand. You have gamma, which is involved in the mitochondrial DNA. So you have all these specialized polymerases that you don't really see in prokaryotes like E. coli, but otherwise the mechanism is still very similar. You just have way more members, way more components to deal with here. So you can see here on figure 9.14, DNA synthesis at the ends of circular and linear chromosomes must differ, obviously. So look at A. Look at 9.14A. This is a circularized chromosome or a plasmid, right? This would be found in prokaryotes or in the mitochondria. What do you have? A single origin of replication. Replication around the circle provides a 3' OH group in front of the primer. Nucleotides can be added to the 3' OH group when the primer is placed. So that's very easy, straightforward. Whereas look at our human or eukaryotic chromosomes. They're linear, right? They have telomeres at the end. And because of that, you have to deal with the telomeres, and it's not trivial dealing with replication of chromosomes with telomeres. And here's why. Let's go through this. Number one, in linear DNA with multiple origins of replication, elongation of DNA in adjacent replicons provides a 3' OH group for replacement of each primer. That makes sense. So you've got primer DNA, until the bubbles, the replication bubbles, bump into one another and fuse. Number two, the terminal primer. This is the one you're dealing with. The terminal primer. The primer at the very end of the chromosome, at the telomere, okay? The terminal primer is positioned 70 to 100 nucleotides from the end of the chromosome. Then it leaves a gap that is not replicated. So you see, there's the terminal primer. You have a gap that is not replicated, and that's a problem, right? Conclusion, in the absence of special mechanisms, if you don't take care of this, take care of this gap here, that's 70 to 100 nucleotides from the end of the chromosome, if you don't take care of that gap, DNA replication would leave gaps at the end of the chromosome, and the chromosome would shorten each time the cell divides. That is a huge problem. You don't want your chromosomes to shorten every time the cell divides, right? And when you have linear chromosomes, that's a true problem. If you don't recall why, again, watch that video from Biology 1406 that makes it clear why the ends of the chromosomes would shorten at the telomeres every time the DNA is copied, every time the cell divides. So what do we do to take care of that? Well, there's a special enzyme involved. Let's look at figure 9.15. The enzyme telomerase is responsible for replication of the chromosomal ends. Without telomerase, your chromosomes would shorten every time the DNA is copied and every time the DNA gets, the cell gets replicated. So take a look here. The telomere has a protruding end with G-rich sequences. So at the telomere, you have a protruding end with G-rich sequences. The enzyme telomerase binds, that this yellow thing is telomerase. It binds, it has embedded inside of it an RNA template that binds to this overhang. The RNA part of telomerase is complementary to the G-rich strand and pairs with it, providing a template for synthesis of copies of new repeats. So you see, you can repair that template strand, you can repair the ends of the chromosomes, the telomere ends. So nucleotides are added to the 3' end of the G-rich strand. After several nucleotides have been added, the RNA template moves along the DNA, more nucleotides are added, and then the telomerase is removed. So you see, you've just extended the telomere. you've repaired it. You've prevented shortening of the telomere ends, the ends of the chromosome. So synthesis takes place on the complementary strand, filling in the gap due to removal of the RNA primer at the end. Conclusion: telomerase, the enzyme telomerase extends DNA, filling in the gap due to the removal of RNA primer. Isn't that cool? So if not for the enzyme telomerase, our chromosomes would shorten every time they're copied. Every time the DNA is copied, our chromosomes would shorten every time the cell cycle progresses. So it's very important that telomerase does its job to prevent the shortening of our chromosomes. And again, prokaryotes don't have this problem because they don't have telomeres to deal with. By the way, telomeres are really interesting. They're implicated in aging. You know, as you age, your cells age and your cells deteriorate. Telomere length has been implicated in that. So reduced amounts of telomerase in aging cells are seen. Conversely, too much polymerase is active in cancer cells. So when cells are dividing too frequently, that's cancer, and that leads to, you know, tumors and metastasis, cancer. There's too much telomerase, keeping the cells, keeping the chromosomes too robust, so the cells can divide too much. In aging, the telomerase is too inactive, so the chromosomes are degrading too fast and shortening too fast. Isn't that interesting? Telomeres are linked to longevity and linked to cancer. It's really interesting field of study as well. So concept check number 7. What would be the result if an organism's telomerase were mutated and non-functional? Oh man. No DNA replication would take place. That doesn't make sense. The DNA polymerase enzyme would stall. No. Chromosomes would shorten. Yep. RNA primers would not be... No, that doesn't make sense. So it's C. Your chromosomes would shorten. And this is what we see during aging. We do see less telomerase activity in aging and shorter chromosomes during aging. Very interesting. So then last concept of the chapter, homologous recombination. Remember that you can have crossover occur, homologous recombination. This involves these structures called Holliday Junctions and single breaks. You can have, this is a homologous chromosome. So this is one homolog in red, and here's the other homologous chromosome in blue. They have areas of homology, which means similar sequences. You then have, okay, so let's look at this. Homologous chromosomes align, and then a single strand break occurs in the same position on both molecules, right? And then you have invasion, what's called strand invasion. A free end of each broken strand migrates to the other DNA molecule. This is called strand invasion. And then each invading strand joins to the broken end on the other DNA molecule, creating a Holliday Junction. That's all a Holliday Junction is. It's this structure here of after the strands have invaded the homologous DNA, creating a Holliday Junction, and begins to displace the original complementary strand. Then branch migration takes place as two nucleotide strands exchange positions, creating the two duplex molecules. So it basically continues copying on. Okay, the crossover moves down. In this view of the structure shows the ends of the two interconnected duplexes pulling away from one another. So you can see the branch point as this crossover progresses. You can see it kind of spread out. And then you can have either a cut along the horizontal plane or the vertical plane. Okay, you could have a cut this way or a cut this way. And depending on which way the cut occurs, if it's a cut across the horizontal plane, cleavage in the horizontal plane results in joining of the nucleotide strands. So you get recombination on this strand in the middle. But a vertical cleavage would give you joining and nucleotide strands where you have the recombination happening on these sides, on the other strands, right? So you don't. This is obviously pretty advanced stuff. You don't need to know this in great detail for my class, but just realize that when there's homologous recombination occurring, this is what's happening. You have strand invasion, a strand nick, strand invasion, the Holliday junctures. Then you have the resolution of those junctures here, and depending on how they cleave, you're going to end up with different types of crossover events, different kinds of recombinants. Okay, so you could see you could have a non-crossover recombinant produces non-crossover recombinants consisting of two heteroduplex molecules, or you can have crossover recombinants produces crossover recombinants consisting of two heteroduplex molecules, different ones. Conclusion: the Holliday model predicts non-crossover or crossover recombinant DNA depending on whether the cleavage was horizontal or vertical, on the horizontal plane or the vertical plane. Again, don't bog yourself down too much with this detail. It is quite complex and beyond the scope of my class. But just realize that this is what I mean by homologous recombination. This is what's going on during that recombination. So again, why is recombination important? Very obviously because you want genetic variation. Remember, during crossover, during crossing over, you want genetic variation. You want that genetic variation to occur so that your offspring have a greater genetic pool, diversity, so that they are more able to persist past insults like selective agents in the world, and that the population can persist, right? So again, really interesting chapter. I hope you learned a lot. Very interesting stuff. Please let me know if you have any questions in the comment box below, and I'll catch you guys next time.
Dr. D, Dr.